Good morning, everyone. Thanks for the introduction. And my name is Vijay Somalinga. I'm a postdoc at NC State, Department of Plant and Microbial Biology. And our lab works on biofuels. So one of the aspects we work on is improving biofuel production in marine microalgae. I'll start off with a very one-slide brief introduction. We all know fossil fuels are not going to last forever. So there are lots of alternatives people are looking at, and one of the alternatives is biofuels. But before going into what I'm doing and how we are trying to improve biofuels, I'm just going to talk to you about the regular gas that we use in our cars, in our planes, in our trucks, and everything. These are all petroleum-based fuels, and these are basically long-chain hydrocarbons. What that means is these are basically chains of hydrogen and carbon put together. So basically, these ones here. How do we get these hydrocarbons? We get these hydrocarbons from petroleum or crude oil by a technique called fractional distillation, which means at different temperatures, you have different hydrocarbons coming out. Hydrocarbons that are 1 to 4 carbon, these are gases such as propane, they come out first. And then they have medium chain hydrocarbons, which are basically liquids, and these are the components of the gas that we use in our cars. And then we have long chain hydrocarbons, they tend to be solids, and these asphalt and fuel oil belong to this group. Like I said earlier, crude oil-based or petroleum-based fossil fuels are not going to last forever. So we have been looking at different avenues for fuels. These include hydrogen, ethanol, natural gas, and biodiesel. So we are particularly concerned about biodiesel. We want to produce biodiesel. This is basically, again, medium chain hydrocarbons. We want to produce these hydrocarbons that can go into a car or an aircraft or a truck, and we are looking at microalgae to produce this. So why are we looking at microalgae? First of all, what is microalgae? What is microalgae? Basically, these are microscopic algae. And I hope you all know what an algae is. Example would be a seaweed. A seaweed is an algae. These are microscopic forms of algae. And the good thing about microalgae is they produce a lot of lipids. So when we say we are producing biofuels from algae, algae doesn't directly produce these biofuels. They actually produce lipids. These are, the way to compare this is, just as we get fat, algae gets fat too. They accumulate these lipids at certain conditions. There are different kinds of plants that produce these lipids, soy, sunflower, castor, but as you can see, algae produces the most lipids. So that's why we are working on microalgae. But the question is, since we already know microalgae produces these lipids and this can be converted into biofuels, what are we doing with this now? Well, one of the biggest drawbacks of microalgal biofuels is the fact that it's expensive to produce. Right now, we don't have the right technology. We don't have established technologies to produce these biofuels in large amounts. So this is where we come in. We are working with different groups, engineers, people from sociology, economists, all these people, and we are trying to come up with a solution to produce large-scale biofuels. So one of the biggest advantage of using microalgae instead of using other crops, for example, castor and sunflower or soybean, is because you can actually grow large amount of algae in small space. These are bioreactors. These are basically long tubes that are transparent, and you can grow algae in these types of bioreactors, or these are called raceway ponds. These are just open ponds that has water, and you put water in it, sunlight, and nutrients, your algae will start growing. So that is one of the biggest advantage of growing microalgae, is that you don't really need expensive setup to grow these microalgae. And like I said, you need sunlight, carbon dioxide, and water, and nutrients. Microalgae will grow, produce lipids, and you basically concentrate it or collect all the algae, get rid of water, squeeze it out, squeeze the oil out. You can convert the oil to hydrocarbons. And so that process, that simple process of converting or collecting the algae and then converting to hydrocarbon, even though it sounds simple, since it's not standardized, it's expensive. So our idea in our lab, we are working on different aspects. We specifically are working on bioengineering this algae by use of genetic engineering, and we want to make sure that these algae produce large amount of these lipids. Another group in the engineering is working on the aspects of the bioreactor. So basically what we are showing here is we are going to have a bioreactor where we can grow the algae. We'll take the lipids out, and then we are going to use the broken down algal cells, recycle it, put it into, again, in the growth chamber, so that we are not wasting anything. So it goes into a cycle. Basically, you can grow the algae, take the lipid out, anything that, all the waste, dead cells can be recycled. In that way, we can actually reduce cost of producing microalgae. So how are we going to make these algae produce more lipids? One way, technically, we have different ways of doing this, but one way is by genetic engineering. So we are going to be using proteins from bacteria. These bacteria are called extremophiles. These bacteria grow in really hot temperatures or really cold temperatures. That's why they're called extremophiles. And we are going to be using a particular enzyme. These are proteins called carbonic anhydrase. And we are going to introduce this protein into algae, and hopefully that will improve the growth of algae and thereby lipid production. So the second step, what we are looking at is biomass recycling. So like I said, we are going to extract the lipids, and then all the rest of the biomass, we are going to break down with bacterial enzymes. These are, again, enzymes from bacteria that live in really high temperatures. The reason we want to use that is because industrial processes tend to have high temperatures, and we need enzymes that are stable at high temperatures, and that's why we're using extremophile enzymes. And then the third step is a one-step bioconversion of the lipids to biofuels. So remember when I said algae produces lipids, they don't directly produce these hydrocarbons, and that's where this step comes in. So we want to convert these microalgal lipids to biofuels, again with the use of bacterial enzymes. The reason we're using a lot of bacterial enzymes is because it's easy to produce and it's easy to manipulate. And these tend to be very, very stable compared to enzymes from other organisms. So why are we using carbonic anhydrase? I assume everybody knows what photosynthesis is, and carbonic anhydrase is one of the important enzymes in photosynthesis. What it does is it converts carbon dioxide to bicarbonate and bicarbonate to carbon dioxide. And previously, people have shown that when you introduced this carbonic anhydrase enzyme in plants, so this is a plant that has the carbonic anhydrase in it, and these two don't have them, and you can already see the difference how well they are growing. So this is the same concept. Since algae are plants too, we want to put these carbonic anhydrases into algae, and hopefully we will see that the algae will grow better, will have more biomass, and thereby they will produce more lipids. So the second step is, like I said, so once we have a bioengineered algae that's producing lots of lipids and growing much better than the wild type or the parent, then we are going to extract the lipid, and then we are going to use enzymes called nucleases and proteases. These enzymes are basically enzymes that will degrade proteins. Proteases degrade proteins, and nucleases degrade DNA. So after you extract the lipids, the leftover that you have is basically composed of DNA, proteins, and other lipids. So we want to break this down into smaller components so that we can recycle this into the growth chamber, thereby reducing waste. So we are going to be using two different bacteria, enzymes from two different bacteria, Geobacillus and sulfhydrogenobium. So these both are organisms that grow at very high temperatures. This one grows at about 60 degrees, and this one grows at about 50 degrees, so Celsius. So they grow at really high temperatures. And we are going to introduce this in Denaliella or Nanochloropsis. These are microalgae that have been shown to produce a lot of lipids. And then after we harvest the lipids, we'll break open them, turn up the heat, and these proteins should be active, and they should degrade the biomass. And the third step is this is the step where we are actually converting the lipids to biofuels. So what the algae produces is this compound here. It's called triglycerides. So you can't put that into a gas tank and make the car run. You have to convert that to a fatty acid or simple fatty acids like this one. And then you have to convert these simple fatty acids into hydrocarbon. That's called biodiesel, or it's just basically a long chain or medium chain hydrocarbons. So to convert the fatty acid to hydrocarbon, what people do now is they use expensive chemical catalysts. I think it's platinum-based catalysts, and they use high temperatures, and these are very expensive processes. So what we are doing is we are using an enzyme. It's a cytochrome P450 based enzyme, and these enzymes we have recently, it's been shown that these enzymes can convert fatty acids into hydrocarbons, and we are working to improve this enzyme so that, and we are also using an enzyme that works at very high temperatures so that these enzymes will be very stable, and we'll be able to convert the fatty acids to hydrocarbons. So basically what we have seen is we are trying to improve the microalgae growth, and then we are trying to use the spent biomass for the bioreactor so that we can improve the cost efficiency. And third, we are trying to use enzymes which will be relatively cheaper than expensive catalysts to convert fatty acids into biofuels. So in future, we are going to transform. When I say transform, we're going to put the carbonic anhydrase I talked about into algae and see how it improves the growth of microalgae. And then we're going to assess how nucleases and proteases convert the biomass, and how we can use these degraded biomass as food supplement for the algal growth. And finally, we are going to analyze the efficiency of cytochrome P450 in converting the fatty acid into hydrocarbons. And basically, I just want to acknowledge everybody who helped me with this work. I'm from Dr. Grundon's lab, and I'm working as a postdoc in her lab. So Hannah is the one who's is a graduate student. She's doing her PhD, and she's working on the last step of the process. I'm helping her out with that. Chloe, Hannah, and Ashika are high school students who actually came to our lab to help me with some of the work I'm doing. And another lab from biochemistry that helped me is Greg, Dr. Rose's lab, and Greg and Paul helped me with some of the other aspects of my research. So if you have any questions, feel free to ask.